right, welcome everyone. Today, we are diving deep into a super high-yield topic for your exams, cholera. Let's get right into it. Okay, let's kick things off with a classic exam question. Picture this. A patient walks in with acute, profuse, watery diarrhea, and they describe it as rice water stool. What's the first thing that should pop into your head? Yep, cholera. By the end of our session today, you'll be able to tackle a question like this with your eyes closed. So here's our game plan. We're going to meet the culprit, Vibrio cholerae, then we'll look at its mechanism of attack, or a pathogenesis. After that, we'll cover the clinical picture, how to crack the case with lab diagnosis, and of course, treatment and prevention. And we'll wrap it all up with a quick revision to lock it all in. First up, let's get to know the bug behind all the trouble, Vibrio cholerae. Okay, so what is this thing? Well, Vibrio cholerae is a curved, comma-shaped bacillus. Think of it like a little comma. It's gram-negative, and this is super important, it's actively motile. It has this single polar flagellum that lets it zip around. Robert Koch first pinned it down way back in 1886. The key takeaways for you are that comma shape and its motility. You'll see why that's so crucial later on. Now, one really cool way we classify Vibrios is based on whether they like salt. You've got the halophilic ones, they require salt to grow, and then you have the non-halophilic ones, which can grow just fine without it. Our guy, Vibrio cholerae, falls into that second group. And here's a fantastic mnemonic for your exams. Think CM for the non-halophilic types. That's Vibrio cholerae and Vibrio mimicus. Just remember, chief ministers have high blood pressure and should avoid salt. Simple, right? Okay, let's talk classification, specifically serogroups. This is all based on something called the O antigen. Now, there are a ton of these, like over 200, but don't panic. You only need to remember 3. First, the big one, O1. This is the main villain, responsible for all 7 global pandemics. All of them. Then you have O139, also known as the Bengal strain, which caused some really nasty epidemics in India and Bangladesh. And finally, there's the non-O1/O139 group. These guys just cause sporadic diarrhea here and there, but they don't cause widespread epidemics. So for the exam, O1 is your pandemic causer. And if we zoom in on that O1 serogroup, we can break it down even further into 2 biotypes, classical and LTOR. And trust me, this table right here, it's a classic exam question waiting to happen. The classical biotype caused the first 6 pandemics, but the current one, the 7th pandemic, is all thanks to the LTOR biotype. And you can see why. It's just tougher. Look at the differences. LTOR is positive for beta hemolysis and the VP test, and it's resistant to polymyxin B and group 4 phage, unlike the classical type. These are the key distinguishing features you'll get asked about. All right, so now that we've met our culprit, let's get into the juicy part. How does this tiny little comma-shaped bacterium cause such a catastrophic disease? Let's look at its mechanism of attack. This diagram is everything when it comes to understanding the pathogenesis of cholera. It looks complicated, but we're going to break it down step by step. We'll see how the cholera toxin basically hijacks our own intestinal cells and turns them into little water pumps. So let's walk through it. First, the toxin's B subunit acts like a key, binding to a specific lock on our intestinal cells called the GM1 ganglioside receptor. Once it's docked, the A subunit, which is the real troublemaker, gets inside the cell. What does it do? It permanently flips the on switch for an enzyme called adenylate cyclase by messing with a G protein. This causes the cell's CMP levels to just skyrocket. This massive spike in CMP sends 2 signals. 1, it tells the cell to stop absorbing sodium. 2, it tells it to start actively pumping out chloride ions. And as you know from basic physiology, where salt goes, water follows. All that water gets pulled from the body into the intestine, leading to that profuse watery diarrhea. Okay. So that's what's happening at the cellular level. Now let's zoom out. What does this look like in an actual patient? What is the clinical picture? Now, here's something that might surprise you. Most people who get infected with cholera feel nothing. A full 75% are completely asymptomatic. Another 20% get a case of mild diarrhea. It's really that last 5% that we're worried about. This is the group that develops what's called cholera gravis, the severe, life-threatening form of the disease. And if there is one thing, one hallmark sign you take away from this, it has to be this: rice water stool. This is the classic description. It's painless, it's incredibly voluminous. We're talking liters and liters, and it has these little flecks of mucus in it that look like grains of rice floating in water. The smell is often described as fishy, but it's not offensive. And a key point, no blood, no pus. This is a fantastic clinical guide because it shows you that everything, all the signs and symptoms, are directly tied to how much fluid the patient has lost. You can see here as the percentage of fluid loss goes up, the signs get more and more severe. Things like decreased skin turgor, a weak pulse, sunken eyes, and if it gets really bad, you're looking at shock and organ failure. It's all about dehydration. So you've got a patient with rice water stool, you're suspecting cholera. How do you actually prove it? Let's move on to the lab diagnosis. Okay, for your practical exams, these two images are gold. On the left, we have a gram stain. See those curved, comma-shaped bacteria all lined up? That's the classic fish in stream appearance. It's beautiful, isn't it? And on the right, this is what it looks like on TCBS agar, which is a selective medium. Vibrio cholerae ferments the sucrose in the agar, and that's what gives you these big, beautiful yellow colonies. You see this? You think cholera. But it's not just about what it looks like. Remember we said it was motile? Well, if you look at it under a microscope using a hanging drop method, you'll see this incredible darting motility. It's not a gentle swim, it's a rapid shooting movement. For culture, we first enriched the sample in alkaline peptone water before plating it on TCBS. And then there's the super cool and simple string test. You take a colony, mix it with a drop of 0.5% sodium deoxycholate, and when you lift your loop, it forms this mucoid string. So cool! And for your biochemicals, remember, it's oxidase positive and indole positive. All right, I know remembering a bunch of biochemical tests can be a pain. So here is a super high-yield mnemonic for you. Just remember, COCUI. That stands for catalase, oxidase, citrate, urease, and indole. And here's the pattern. It's positive, positive, variable, negative, positive. So catalase a positive, oxidase positive, citrate can be either, urease is negative, and indole is positive. COCUI. Lock it in. Okay, we've identified the culprit and we know how it works. Now for arguably the most important part, what do we do about it? Let's talk treatment and prevention. Let me be crystal clear about this because it's the single most important message for treating cholera. The cornerstone of treatment is not antibiotics, it is fluid replacement. Prompt, aggressive rehydration is what saves lives, period. For mild to moderate cases, we can use oral rehydration solution, or ORS. But for a severely dehydrated patient, you need to get an IV line in immediately and start them on Ringer's lactate. So that's how we treat an individual. But how do we prevent this from happening in the first place? Well, on a larger scale, it all comes down to basic public health. Safe water, proper sanitation, and good food hygiene are the ultimate weapons against cholera. We also have oral cholera vaccines, which the WHO recommends using during outbreaks to get them under control. And finally, and this is crucial, cholera is a notifiable disease. That means every single case has to be reported so public health teams can jump into action and stop it from spreading. Okay, deep breath. We've covered a lot. Let's do a quick rapid-fire revision to make sure all the most important points are stuck in your brain for exam day. If you walk away with just 5 things, make it these. 1. The villain. It's Vibrio cholerae, that gram-negative, comma-shaped motile bug. 2. Its weapon. The cholera toxin, which sends CAMP levels through the roof. 3. The crime scene. That classic rice water stool causing severe dehydration. 4. The evidence. Look for that darting motility and those bright yellow colonies on TCBS agar. And finally, number 5, the solution. Rehydration is number 1. That's it. That's the core of cholera. And I'll leave you with one final thought to chew on. We know that Vibrio cholerae naturally lives out in the environment, in marine ecosystems, often attached to things like crustaceans. So, as our oceans get warmer due to climate change, what do you think that means for the future of cholera outbreaks? How might that change the way we have to fight this ancient disease? Something to think about.